If you already knew the full sequence of the human genome and tried to write it down by hand at a rate of 1 nucleotide per second, it would take you nearly 6 billion seconds, which is approximately 200 years to finish. With current technologies, however, such as NGS, we could sequence the human genome in about a day. How is that even possible? How could we condense 200 years of work into one single day? Well, it's all thanks to modern molecular techniques in bioinformatics. But what even is bioinformatics? How does it help us accomplish such fascinating tasks? Rather than a dry definition, let's take a history ride together and explore what bioinformatics is. The first sequencing method, termed Sanger sequencing, was inspired by DNA replication itself, but with Sanger's genius twist. In normal DNA replication, a short piece of RNA called a primer attaches to a single strand of DNA and serves as a starting point. Deoxynucleotide triphosphates join the growing chain via their phosphate groups. Notice, a free OH group at the end of the growing chain is necessary for nucleotide addition. The modern method of Sanger sequencing provides the replication complex with a blend of normal nucleotides and D-deoxynucleotide triphosphates, which have lost their OH and are also fluorescently tagged. Whenever a ddNTP is incorporated into the chain, it will terminate replication. Well, because that chemical bond that attaches two nucleotides together is catalyzed between the phosphate group of one nucleotide and the hydroxyl group of another. Without the hydroxyl group, incorporation of new nucleotides would become impossible. Multiple replication cycles are carried out during Sanger sequencing, and as we progress from one replication cycle to another, random incorporation of ddNTPs results in a collection of DNA fragments with different lengths. At the end of the last cycle, we collect these DNA fragments and run them through a gel, where smaller fragments go faster, thus arranging them. A laser then detects the fluorescent tag at the end of each fragment. By lining up the signals from shortest to longest, the full DNA sequence is constructed. With the progress made in the field of genome sequencing, biologists began planning to use these techniques to sequence the whole human genome. As a result, the Human Genome Project launched at 1990. For the first time, human beings were on a quest to unravel the mysteries hidden within their own cells. A project so big, some call it the Apollo of biology. So could Sanger sequencing really do the job? Sanger sequencing was great, but had one big limitation. Sanger sequencing could reliably read a DNA segment of 600 to 1,000 nucleotides long. The human genome? Well, it's about 6 million, billion nucleotides. Biologists' solution was to use restriction enzymes to break the human genome to small manageable fragments Sanger sequencing could handle. Now, could they sequence each segment? There is also another problem that we have to somehow solve. The quantity of every individual DNA fragment was also not sufficient for sequencing, so they had to do something. What did they do? Scientists inserted each fragment into plasmids and then transfected bacteria, cultured bacteria. And then, as bacteria replicated, they amplified our plasmid as well. So now each segment could be sequenced after amplification. Now we have the sequence of all of these fragments. How are we going to arrange them in the right way? It's almost like a puzzle with 45,000 pieces. How could we solve this puzzle? Where does each piece fit? Well, that is where bioinformatics comes in. This massive amount of biological data must be analyzed some way to actually obtain the big picture, but how? I will try to give you a simplified explanation. Look at these two sequences. When carefully observed, their overlaps become obvious, leading us to a solution of their full-length sequence. In the Human Genome Project, the digestion of the human genome was carried out in such a way that small fragments would have overlaps. Using computer science and the power of algorithms, all these 45,000 segments were arranged in the right order to solve this massive puzzle. In the big picture, the human genome. After 13 years of hard work, at least 20 laboratories, institutions, and companies at the cost of $3 billion, humans eventually discovered the human genome for the first time. The Human Genome Project certainly wasn't the last time biological data was produced. Each and every day, 5 to 10 petabytes of genomic data is produced. How much data is that? To imagine it, approximately 3.5 million computers are required to store accumulated biological data of 1 year. Of course, storing this amount of data is itself a challenge. Servers store this great amount of data in an organized manner. That's why we call them databases. Every researcher could send requests to these databases via internet and obtain whatever data they need. It's almost like a librarian. You give them a query and they search for books that match your query. So let's get back to the earlier question. What is bioinformatics? We just saw what bioinformatics was. It analyzed a great deal of biological data. It manages and stores data, manipulates it. With what? With computers, because humans can't analyze that great amount of data manually. What is bioinformatics? Bioinformatics is an interdisciplinary field of science that aims to use computers to manage, store, and analyze biological data. The Human Genome Project and storage of biological data, of that particular biological data, are just a few examples of the applications of bioinformatics.